Hello, everybody. This is CaveNet, and it says webinar series. And tonight we have part 3 from Dr. Hazel Barton, our well-known caver. Again, I want to talk about Hazel. We can't thank her enough for her taking the time to help us and give us this information that we really can't get anyplace else from someone that we trust. But I'm going to give you a little background on Hazel, a little because she doesn't give me much. There's so much I could say, but she doesn't give me a whole lot. Anyway, Hazel has a PhD in medical microbiology, immunology. She is a professor of biology and geology and has been teaching medical microbiology for over 20 years at the undergraduate and graduate level. Hazel served 6 years on the Public and Scientific Affairs Board of the American Society of Microbiology and is a fellow of American Academy of Microbiology. She's also a very well-known caver. She caves worldwide, has an amazing resume, and has been in just caves that are stunning. But what I'm going to do now is I'm going to go ahead and turn this over to Hazel so that she can go ahead and start, and so that we don't waste her valuable time. But we really appreciate you being here, Hazel. So go ahead and unmute yourself and let's get going.
Anyway, thanks, Debbie. Thanks for the introduction. Appreciate it. Thanks everyone for coming back. These things can get a bit daunting, and I try to keep it fairly simple. If anybody wants to pursue this this subject matter, give me an email. I can send you some papers. Just to reiterate, I don't say anything in here that I can't back up with peer-reviewed published material. So if I say, a percentage of people have been shown to this, I've actually gotten the paper, found the paper, read the paper, and pulled that out. And I've got all the references for that in there. So there's too much information of people putting stuff online saying, I heard this and I heard that. So just to reiterate, there's nothing I'm presenting that's not being published. It's been really hard keeping up with the literature. I believe by the beginning of June, there had been 24,000 papers published on COVID-19 and SARS-CoV-2. And I think the publication rate is about 4,000 papers per week. So it's really difficult to curate and find the important ones. What I try to do is just focus on papers by people who are experts in the field, who publish in good journals, and have data that you can actually analyze and look at yourself. So today I'm going to talk about trying to get ahead of coronavirus. So this is not going to be reiterating anything that I went through in the past coronavirus lectures. So if you haven't seen those before, you might want to stop and go back and look at those before you listen to this one, because I'm not going to explain too much about what the virus is or how it makes you sick. Those are in the past webinars. If anything's changed significantly, then I'll give you that information in this webinar. So I'm going to focus on antiviral agents, so drugs that can be given to people who are sick, and then vaccines and where we're in vaccine development. So I'm going to talk today about the current statistics. So what do we know about the disease now that we didn't know a month ago? The state of antivirals, and how antivirals work actually. Immunity and vaccine. I did actually start to put together an immunity presentation, and I realized I probably spent 4 hours trying to explain the immunity before I got to the vaccine part. So I very much simplified that, but if anybody wants me to do just in a immunity webinar, I can do that. And then unfortunately, I'm going to have to talk about bats again. There's been a few papers that people have brought my attention to, and they're confused what that means about spillback and bats in North America. So I'm going to talk to that again. Current statistics, confirmed cases worldwide. Every time I go to write a paper and it's gone through review, I have to change the number of cases by at least 1 million. So we're up to 1 million cases worldwide, over 400,000 deaths. So it's kind of hard to tell where we're at with the total percentage mortality, but it's not good. 400,000 people is not what you want to see. And we haven't got a handle on this yet. This disease is still continuing. So what do we know this month that we didn't know last month? So 10% of all cases of COVID, 10% of cases of COVID cause 80% of infections. So what does that mean? That means if you have 100 people in a room, 10 of the people in that room are going to cause 80% of the infections. So we think that there are certain people that spread a lot of virus. So a lot of people get sick, they don't spread the virus, they don't make other people around them sick, but we do get these super spreaders. So 10% of people who have the disease, or 10% of cases, cause 80% of infections. So people that are subsequently going to get the disease. 79% of infections are caused by asymptomatic people. I know there was some confusing data today out from the World Health Organization saying that asymptomatic people didn't cause disease. That's certainly not the case. It means that the people who cause most of these infections don't realize that they're sick. They may have very mild symptoms. They don't understand that they are sick from those symptoms. And those things can be, losing your sense of smell, your GI tract. I mean, really, really subtle things that on a day-to-day basis wouldn't think that you're infected with the coronavirus. The chances of catching the coronavirus if you are exposed to somebody for 10 minutes without wearing a face mask within less than 6 feet is about 15%. So I just want everyone to remember that 15% chance of infection, because I'm going to bring it up again. So you're not guaranteed that you're going to catch the infection, but if you are around a person for 10 minutes without any kind of protection and when you're within 6 feet, there's a 15% chance of infection. And obviously, if you're around a super spreader, that might go up. And if you're around somebody who's not going to spread a lot of virus, that might go down. So we now know that there are speech super emitters. So there are people that produce a lot of virus particles. Some of these people can produce 100,000 particles an hour. We don't know how much virus you have to make you sick, whether that's just one virus particle, a couple of hundred, or a few thousand. But there are certain people that spread a lot. It's not based on gender. It's not based on, vocal cords. There's no anatomy. It's probably a lot to do with the virus itself rather than the anatomy of the person who's doing it. It tends to be emphasized by anything that causes you to raise your voice or resonate. So there was an event, there was a choir that got together and sang for 2 hours, and 87% of the people in that choir in 2 hours got the infection from one person. So it's thought that singing, projecting your voice, exercising, all those activities actually might increase the likelihood of spreading the virus. 65% of cases are from direct person-to-person contact. So that means that the idea of fomites, so the idea of picking up from door handles or surfaces, is actually not as important as we once thought it was. We thought it might be the primary way that the virus is passed, but it's actually it's direct contact between people, either talking to each other or touching. And I've seen that number go as high as 87%. But the only published peer-reviewed study I saw was 65%, but I have seen that as high as 87%. The chance of getting an infection indoors is 19 times higher than if you're outside. So if you're inside in a poorly ventilated room with the same person standing next to them versus if you were outside with the same person, you are 19 times more likely to catch the virus. So most people are catching it from each other indoors. And there seems to be an issue for some people with clearing the virus. So some people are getting infected and there's still detectable virus. So you can hear cases where somebody goes into the hospital and they're in hospital for 6 weeks. There are now some cases of people that are ending up in hospital and still producing the virus 10 or 12 weeks after the infection. Now that's based on the genetic material of the virus, and the studies to determine if they're actually making infectious virus are ongoing. But it does appear that some people really have a hard time with their immune system clearing it. So these are just the references I used to pull that information. Masks have been shown to be very effective. There was a recent study that showed that if somebody who had the virus wore a mask, they reduced the likelihood of producing infectious particles almost to zero. So if you are out and about and you're not wearing a mask, and there's somebody out and about who is infected and not wearing a mask, then they are likely to spread the virus. There's the idea of this social distancing, and this 6-foot distance is based on the idea of droplet transmission. But there's new data that suggests if you're in low humidity, those droplets will actually dry out quite quickly. Within less than a second, those droplets that are coming out will dry. And as they dry, they get smaller. As they get smaller, they get buoyant, and they can actually float in the air for up to 30 feet. So once we thought as long as you had social distancing, then you didn't really need to worry about that mask, but it appears that masks are really critical. So they've really improved reducing the droplet and aerosol transmission. And I said a super spreader can produce 100,000 virus particles an hour, and it could be as low as 1 virus to make you sick. What hasn't changed? The R0 value. So if you remember, this is the value for how many people a single individual on average infects. So that remains between 2 and 3. It seems that it's patchy though. So what you tend to see is you might have one person that infects 50 people and then 10 people that don't infect anyone. So there's a patchiness to that R0 value. So it's not as straightforward as every person who's infected is going to infect 2 to 3 other people. The more that a state implements social distancing and mask wearing, the more that R0 value is dropping. Most people will shed virus. This is done based on infectious particles rather than just genetic material. But most people, once they get sick and start showing symptoms, they are shedding the virus for about 5 to 10 days, and then they stop doing it. And then the mortality rate still continues to hover around 1%. So anyone that tells you what that means that there's 99.9% recovery, well, the percentages are off, but 1% is still a really big number. When you think about the size of the population across the US, you're looking at 30 to 40 million people dying if we don't limit the infection. And it's not even. That's even if you take into account all age groups. So if you take into account from 0 to 100 years, you average that out at 1%, but it changes dramatically if you are in different age groups. So this is a graph from Italy with total deaths above people aged 30, and there were 6,700 deaths, and I think I wrote the percentage down here. Yeah, 83% of those deaths were in people above the age of 70. So just because it's 1% for the general population, if you're in a higher risk group, don't assume that it's 1%. It could be as high as 30, 35 to 40% for your age group. Okay, any questions? I don't hear any, so I'll keep going.
I'm sorry, I didn't unmute the right thing.
Okay.
One question, it says, so do we need 30 feet spacing in a cave with high humidity?
High humidity seems to denature the virus, so virus particles will die more quickly above 50% humidity, but I wouldn't assume that that makes it safe to be in a cave. You should still practice social distancing and wearing masks. I think if you are going to be caving, you might want to think it creating cohorts, so people that function much like a family unit where they only socialize together, and that makes it a lot easier for contact tracing. But the virus particles yes, that's a good point. The virus particles are not as infectious at high humidity, and the humidity itself seems to denature the virus particle to some extent.
Another question is, are super spreaders called that because of the behavior of the viral load?
Yeah, it's just basically we have no idea what makes someone a super spreader. There's nothing about genetics, there's nothing about gender, there's nothing about, size. Some people just happen to do this, and it's probably a lot of function to do with the virus and the immune system of the person who becomes a super spreader. And there's nothing we can do at this point in time to predict what that might be. We might get to the bottom of it eventually, but at this point, it's super spreaders are known in other infections, like white nose syndrome has some super spreader events, but we're not entirely sure for a lot of these diseases how that happens, and we certainly don't know for COVID right now.
Okay, so it's not their behavior, right?
No, there's nothing specific that these people are doing that makes them super spreaders. No, unless you go singing, unless you're going to go stand in a room full of people and sing at the top of your voice.
Yeah, I read that about that. That was very sad. Okay, next one is any differences between masks, and do you have a study on the effectiveness?
Yeah, there's been some studies out there that looked at virus particle load and masks. Most of the studies seem to suggest that wearing the mask protects other people. So you're not wearing it to protect yourself, you're wearing it to protect others. And the greatest effect is if you're wearing a mask, you're not going to spread that virus to other people. Even if you don't think you're infected, you're protecting other people. And so I see a lot of people saying, I can't be expected to wear a mask, and it's not required in some states, and they're going into stores and people aren't saying them and they're harassing them. And it's kind of like, we're talking about the safety of other Americans, and there's people going overseas and fighting in wars to protect our safety, and you can't put a mask on. It's like just get on with it. So yes, masks are important, particularly if you have the virus. You don't know that you have the virus, and therefore it's important for everybody to wear them. If you can get a surgical mask, that's okay, but there's such a limit on them right now. We try to discourage people from using them because they really need to be going to healthcare settings where they're needed. A double cloth mask seems to work quite well.
Okay, the last question is, a hot, humid room like a wet sauna, would this be worse or better or no different?
So it's going to be better. Yeah, it's going to be better because high humidity inactivates the virus, but you don't know how much virus is in the air, and you don't know if you're in the room with a super spreader. So just assuming that maybe if I'm in a sauna, I'm safe, that's kind of a false equivalency because you don't actually know what danger you're in. So let's say you took an average room with an average load, then an average room with average humidity and average load, yes, you'd be safer in a sauna, but you don't know about what the other conditions would be. So don't assume just because you have high humidity, it makes you safe.
And another one says, is an N95 with an exhale valve not a good idea?
Yeah, so the exhale valves tend to allow the virus particles out. They're better than nothing. But if you have a valve on there, you're not really, it's still possible that you're going to be pushing the virus out. So if you're in a healthcare setting and you're going to be wearing a mask all day, you need to be protecting yourself from other people. So an N95 works. It works much better than a regular cloth mask, allowing the virus in if you have a fitted N95. So if you're in a healthcare setting, you should be wearing one of these. But I've been telling people to avoid using the masks with the venting capacity on it because it potentially allows the viral particles out. I haven't seen a study where they've compared that directly to see how that's different.
Okay, that was our last question for the moment.
Okay, so I'm going to go on to antivirals. So I've got a picture of human cells here. So these look like human fibroblasts, and they're actually stained with different dyes that highlight different parts of the cell. And just to give you a little bit of background on the structure of, oops, I'm going to have it looks like I'm not going to be able to use my pen on this today. So there are a nucleus here, and then this is the cytoplasm. And then there's pink stuff in here. This is where proteins are manufactured in the cell. If you remember from your biology, this is the endoplasmic reticulum. And then these little green dots around here, these are mitochondria. So those are the power plants of the cell. So basically, think about a cell as a big bag of cytoplasmic material with a pot of genetic information in the middle, of sac of genetic information, which is where the DNA is stored, and that sends out signals to the protein factories to make proteins. So this is just the structure here. Oh, looks like I found my pen again. All right. So you have a nucleus. So this is going to be the edge of the nucleus here. You have DNA. DNA is translated into a message, and that's called messenger RNA because it's a different structural molecule. So DNA is double-stranded. It's that helical structure. And then messenger RNA is just a code that's read off the DNA that comes out of the nucleus, and then it goes to the ribosome, which is that protein factory. And the ribosome will basically read that genetic code and build proteins from amino acids. And those proteins are chains of these amino acids that fold into different structures. So if you think about all the different structures we have in our cells, they're either made from proteins or they're made from proteins synthesizing other things. So things like lipids and sugars in your cell are going to be made by proteins. So this is a pretty standard mechanism for all life like us, which is we have double-stranded DNA that gets made into a message, which is a single-stranded RNA, and then that goes to a protein. So that's how our cells work. So DNA is deoxyribonucleic acid, and RNA is ribonucleic acid. And this is just to do with how they form their structure. So they have a slightly different chemistry, and that's why one is called DNA and RNA. So all cells have this pattern. They can't deviate from that pattern. So if there is a deviation from it, it's not standard in the cell. So this is the, and I know this is a complicated picture, this is actually the mechanism of replication for coronavirus. So the coronavirus, it sticks to a receptor on the surface called ACE2. It causes the cell to kind of create a pocket that pulls the virus inside, and this is called an endosome. So it makes this endosome. Another way that it can get inside the cell is it can actually directly fuse to the membrane. We don't think that this method is actually important in COVID-19. It doesn't seem to be as important as the ACE2, but it is a mechanism that coronaviruses can get inside. Once they're inside, the virus can change the structure of this endosome. So this endosome is basically like I don't know, just a bubble inside the cell that carries the virus. It changes the structure of it, which causes the membrane of the virus to fuse with the membrane of the cell, the endosome, and then the viral genetic material comes flying out. That viral genetic material is actually an RNA itself. So then those ribosomes, so talked about ribosomes making proteins before, those cellular ribosomes bind to it because they think it came from the nucleus, and they start making copies of the proteins based on this RNA. So you get a polypeptide or a protein chain that is made. Then this protein is made as a single strand of amino acids that have to be chopped up into smaller pieces that are functional. So that gets all cleaved. That cleavage is carried out by something called a protease. Those small peptides and proteins actually go into an endosomal compartment, and they start making more copies of the RNA. So you had RNA come into the cell thought that came from the nucleus, and then the virus is now going to make a protein that looks like a nuclear protein to make more copies of the RNA. All right, so you've gone from an RNA here to an RNA there. And when you do this, you actually get a hybrid. You actually get two strands of RNA because one is serving as a template and one is being made on it. So you can see you've got your template strand here, and then you've got your other strand being formed on it. So you actually have double-stranded RNA. These vesicles then travel to the surface of the cell, and then they fuse with it, and then they release virus into the outside of the cell. So how does that allow us to develop antivirals? Well, what we can do is we can think about ways that the virus behaves this way that's not seen in our cells. So one example of that is how this endosome fuses. So we don't have a process for bringing proteins into to fuse with an endosome like that. So there are actually antiviral agents that actually stop that fusion process. So hydroxychloroquine was one you may have heard of. Then you have these proteases that actually break down this really long peptide. It's called a polypeptide that is made by the virus that needs to be cut. Those are proteases. So it's a protein cleaver, so it's a protease. So you can actually have an inhibitor of that. So if you're familiar with HIV, they have protease cocktails. And that's actually how we treat HIV, is we produce a lot of molecules that actually stop the virus from being able to cleave this polypeptide. And as a result, it can't make anything functional. The other thing we can do is we can target the synthesis of this double-stranded RNA. This is a big cellular target that signals the cell that's infected, but it also has to bring its own RNA replication. So remember, if this is the nucleus here, in the nucleus we have to go from DNA to RNA. And that's the only way it can happen, DNA to RNA. What we're doing here in the cytoplasm is we're making RNA to RNA, and that is not natural. The cell has no capacity to do that. So the virus has to make that protein. So that's called an RNA-dependent RNA polymerase, and it also has to make it in the cytoplasm. And because this structure doesn't exist in any of our cells, what we can do is we can actually interfere with it. We can actually stick something in and interfere with that protein. And if you're familiar with HIV, back in the early days when they made AZT, that was the first. It's called a nucleoside analog because it sticks into the RNA and confuses the polymerase. So it actually confuses the ability of the cell to make the RNA. So you can't make a functional virus. The other couple of things on here, there are neutralizing antibodies. So there's a couple of ways that you can do this. You can actually, your immune system, once you've been infected or you've had a vaccine, your immune system will actually cover the surface of the virus. You can see it here, all the receptors on the surface that the virus would need to attach to the surface, they've been bound by a antibody so that it can't be recognized by the cell. So you can block this either through making synthetic antibodies, which is one method that would be considered an antiviral approach, and the other obviously is immunity. So these are all targets in our cells that aren't natural, and because they're not natural, you can make compounds that are specifically geared to interrupt that function without harming our own cells. So one of the ones we're going to talk about is remdesivir. Remdesivir, this is one you've probably heard a lot about in the news. This is the one that actually blocks the polymerase. So it's blocking that ability of the virus to go from RNA to RNA. The way that it works is that if you think about genetic material, so you're thinking about DNA, remember there's 4 bases. You may have heard of GATC. If you've ever seen the movie Gattaca, that's where it came from. So GATC are the building blocks of DNA. So this makes up our signal. I've put the T in brackets here because in RNA, we actually use a uracil, a slightly different structure, only because it's RNA. So it's in brackets there, but the similarities are pretty much the same. So adenosine is the nucleotide that's normally used when you're making DNA and RNA, and it has this structure. The RNA polymerase, the protein that the virus uses to make more copies of itself as an RNA, is actually slightly different in structure than our RNA polymerases. And because of that, we can actually fool it into taking up something it shouldn't. So with remdesivir, this is the drug. It's injected into the body. It has to go in through intravenous injection. Inside the body, within the cells, it gets converted into this structure, which you can see. You have this oxygen here, you have the ribose molecule here, you have the nitrogen and the cyclic structure here. It looks an awful lot like adenosine, but it actually binds, because of these additions, it actually binds more tightly to the RNA polymerase that the virus uses than the one that our cell uses. So when the virus is pulling things in to make copies of itself, it's much more inclined to pick up the remdesivir version that it is the natural cell version. And when it does that, it can't elongate. Normally what would happen is you'd have an elongation structure along here, and that can't happen because the the nucleoside, the remdesivir, is the wrong shape. So it actually stops and confuses the virus. Now, it doesn't seem to be completely inhibiting the virus, as we've seen in other viruses, but it does slow it down. This drug was actually developed for use in Ebola, but it didn't work great in Ebola. There were 3 drugs that were tested in clinical trials for Ebola, and this one didn't do a good job. And it turns out that somebody tested it in a SARS-like virus and showed that it did have an effect in coronaviruses. So it was used. And there's been a lot of discussion of this. I noticed in the news, there was one study came out that said it doesn't have any effect, and another study that came out said it does have an effect, and everybody was confused. Does it do anything? It doesn't do anything. So there are 2 studies that this is based on. One was done by Wang and colleagues, and the other was done by Beigel and colleagues. The Wang study included 237 patients, and the remdesivir was given at 12 days. And this is a plot that shows, it's a measure of how much better people did. So remdesivir is here, and people who received no treatment is in blue. And you can see that there's really not much difference between them. There's just a subtle improvement. In some places you don't see it. So they concluded that there was no significant improvement for patients that received remdesivir at 12 days. This is 12 days after these patients started feeling sick. They're nearly all in the intensive care or in hospital. These are very, very sick people. The Beigel study was much bigger. It was 1,000 patients, and they started the drug 9 days. So they started 3 days earlier. So this is 9 days after signs and symptoms. And they stopped that study early because they actually noticed a significant difference between the 2 groups, and they realized that they saw what they believed was enough of an improvement that it could be used as a drug. And so that's why one study came out and said remdesivir doesn't do anything, and another study came out and said it does. And it's probably a lot to do with when the drug was actually given to people. So this is a picture of a cell. I think this is a velo cell infected with SARS-CoV-2. So the cell is in blue and the virus particles are in yellow. And you can see that this virus is budding out all over the surface of the cell. And coronavirus, the COVID infection we talked about the last lecture I gave, it's not really a respiratory tract infection. It's a disease that gets in through the respiratory tract that causes a lot of vascular problems. It seems like it's a vascular disease, so it causes most of the signs and symptoms within the blood, and leads to a lot of problems that end up with this huge immune response. And most of the damage we're seeing is actually the body's immune response to the virus itself. So if you've got a cell that's producing this many virus particles, you can imagine that that's triggering the immune system to fly off all over the place. So as a result, it doesn't really help for you to get these antiviral drugs that slow down replication if you're seeing a patient get the drug if they're 9 or 12 days out and they're already in the hospital. And it turns out that nearly all antivirals work if you give them really, really early. So as soon as the person's got that first sign of symptom, or even if you know that they've got a possible exposure, you can start giving them the drug immediately post-exposure. That's when you tend to see treatments. So the idea of having an antiviral drug that you can give to a patient who's in intensive care and you can resuscitate them, that's just not a reality. What you have to do is you have to control the immune response. So the reason that the responses you got to see with these two studies for remdesivir were so, not, I mean, the second study was statistically significant, but it was just a small difference. It's because these patients were so far along with their infection. It's basically they weren't clearing the infection, they were getting sicker and sicker. As a result, they tried these drugs. But you really need to get the drugs into someone very early on. And that gets to another drug, which you've probably heard a lot about, and that's hydroxychloroquine. So hydroxychloroquine works at the stage where the virus in the endosome needs to fuse to the endosome, the membrane of the endosome, like this soap bubble, and pop out into the cell. And it does that by changing the internal chemistry and making it a lot more acidic. That acidity changes the structure of the spike protein, so the surface receptor of the virus. That changes it and allows it to fuse with this membrane. What hydroxychloroquine does is that it actually prevents this endosome from becoming acidified. So it stops it from getting acidic. If it can't get acidic, the spike protein can't change. If the spike protein can't change, then the virus can't fuse and get out. So this is called a fusion inhibitor, and this is what hydroxychloroquine is. So there was a study that showed there didn't appear to be much impact of hydroxychloroquine in the treatment of COVID-19, and you may have heard about this one. This is the big World Health Organization study. It's been going back and forth. Did they do it right? Not do it right? This is the data. So this is basically one is your average likelihood of dying from a COVID infection. So if you're on this side of the chart, you see an improvement, and if you're on this side of the chart, you don't see an improvement. So this is just showing a lot of these comorbidities, so things that you're doing that are increasing your likelihood of getting sick. So coronary artery disease, congested heart failure, diabetes is a big one. Then we know that we have things like ACE inhibitors. They can actually reduce the likelihood of you developing a severe infection. And then below this line here are the treatments of patients with chloroquine. And you can see that giving them chloroquine actually increased the likelihood of their disease progressing to death. So they actually stopped the study early because there was such a big impact of using hydroxychloroquine versus these are all other drugs that have been proposed to use. And you see most of them cause some kind of improvement, but there's a clear shift from using hydroxychloroquine. Now, this was a study of 96,000 patients, and people had a problem with this study because they didn't have any information on where the patients were. So were these patients that were getting hydroxychloroquine early? Were they getting it too late? How long had they been hospitalized? And it turned out that the people who had provided the data did not actually have a background in this kind of delivery of data, and there were some concerns about where that data came from. So this study is being reinvestigated. So the investigators are going back to the original hospitals that provided or presumed to provide the information for this study, and they're getting that information directly as opposed to going through this company. So it's unclear what the company did, where it was just poor record keeping, or they were taking advantage of a situation to provide something that somebody wanted at cost. Now, you would say, oh, that's such a terrible waste because now we don't know if hydroxychloroquine works, but a study just came out last week. This is a much better study. It's a smaller group. It's 821 patients, but it was done within 4 days of exposure. So this is given to people from 0 to 4 days, 0 being that they knew they had an exposure. All of these people received the drug hydroxychloroquine before they developed any signs or symptoms. So it was given to people who had known contact. So they'd actually interacted with people who'd had COVID-19. And as you can see, the hydroxychloroquine is the square and the placebo, so something that's just basically a sugar pill to see if there's an effect. There's no difference between the hydroxychloroquine and the placebo. These are the error bars, right? So this is your statistical significance. So if they overlap, there's no statistical significance there. So this suggests that hydroxychloroquine, even if you were able to give it to people very early, has no impact on the duration or severity of the disease. The last one I'm going to talk about, because I know this is pretty complex, is ritonavir and lopinavir. These are protease inhibitors. So these are actually drugs that are used in HIV. So these are part of the protease inhibitor family of drugs that is used successfully to treat HIV. And they've been used because they stop this long polypeptide getting cleaved into functional proteins. There was a study that was recently done on 200 patients. Again, we don't know where these patients are in their infectious cycle. We don't know how long they've been in the hospital. We don't know what kind of condition they're in. But what we do see is that there's no significant difference between the number of patients that are receiving a placebo and the number of patients on the protease inhibitors. So I think there's something like 90 other drugs that are currently being investigated. So I just highlighted one here, and one here. There's multiples at this point, other protease inhibitors on the surface. There are other things that affect the lipids in the cell. So there's lots of places we can investigate. The interesting thing is that we've already got, one pretty good functional drug. The immune modulators seem to be working really, really well. The IL-6 inhibitor works really well, so that is what turns down the immune response. So we have some pretty good treatments for people already. Not fantastic. There's no magic bullet against there's no magic bullet against viruses. The magic bullet is a vaccine. But we certainly have got something, and it's important to remember with HIV, it took 12 years to get from discovering the virus to the point where we had a stable mix of antiviral compounds that could actually help people survive the disease. So we're actually, this thing is accelerating pretty quickly. So we've got lots of targets left. All right, so any questions?
Yep, there sure are. All right, our first one, let me scroll up there. You said the fatality rate is 1.17%. Is this the case fatality rate or the infection rate? If you quoted the case fatality rate, do you have an estimate on the infection fatality rate?
So this is only people that have been diagnosed with COVID-19. So they've had a test that have demonstrated that they have the virus, or they've had a serology that showed them that they have survived the virus. So we can't estimate what those numbers are. Obviously, if the serology suggests that lots more people in the United States have been infected, then that percentage is going to go down. In some countries, the percentage looks terrible because they aren't testing enough. But until we get serology rolling out, we're not really going to fully understand what the mortality rate is. It is important to remember that that is different based on your age category. So it's really critical that if you're in a high risk category, you need to be really careful.
Let's see, is there talk of using targeted microRNA or other cutting edge treatment to interfere with the replication of protease machinery? And this is from Eric.
Yeah, I don't think that doing like a CRISPR type approach is viable, because you'd have to get that into all the infected cells. So immunity works great because you can get some messenger RNA in and you can get a protein made and you get an antibody response. And it doesn't matter where in the body you have that infection, the antibodies will kind of single it out and destroy it. The trouble with using something like an interfering RNA approach is that you have to target it into all the cells that are either infected or potentially infected. And we don't have delivery mechanisms that really work well enough to deliver nucleic acid material to cells like that. Now, I'm sure somebody's working on it, but in theory, that's very, very challenging.
Another one is, do you know why and I had trouble with this word last time, remdesivir, why it is not used on the first day of symptoms when people are in the hospital?
So it is usually used on the first day of symptoms in the hospital. That's why you're looking at people between 9 and 12 days, because they cannot give it to people unless they're hospitalized. So you can't give it to someone prophylactically. So if they know they've had a contact, because it's an incredibly expensive drug, and it can only be administered intravenously. So you can't have it as an over-the-counter pill. I'm sure if it was available as an over-the-counter pill, then it would certainly be available to people who had known contacts, but you have to be in a hospital environment, and you have to be on an IV for it to be administered. It can't be taken orally. Now that's different from hydroxychloroquine. That is a pill, so you can take that. So that's why they've been able to do those studies. But yeah, I mean, it's a good thought, and it's a good suggestion, but unfortunately it's not practical from a, you know, synthesizing enough of it, having enough available, the cost, and also the fact that you actually have to be on an IV to receive it. But it's a good point. It would be lovely if we could do that.
What are the side effects?
Yeah, they're pretty bad. I'm trying to remember what percentage. I think 8% of people left the study because of the side effects. I'm sorry I didn't make notes on what those side effects were, like nausea, I think, headaches, aches and pains. I think it was about 8% left the study. You know, if it's a drug of last resort, you might be willing to tolerate this, and you may be unconscious as well, so you might not notice those things. But it wasn't as bad as the hydroxychloroquine. I think it was a really high percentage of people that left that. Trying to remember the number. I'm not going to guess, but it was in the teens, maybe 14% of people that left the hydroxychloroquine because of side effects.
Do you know what those side effects were?
I'm trying to remember. I think shortness of breath. There were some heart issues. Again, I'm sorry, I didn't write those down.
Okay. So if somebody already has a heart problem, that would not be something they would want to take.
That's something they'd have to discuss with their doctor. I mean, that's a conversation between them and their doctor, yeah.
Another one is, what might account for the hugely different severities of illnesses in different ages, especially very young people? I think we addressed that on the last webinar.
Yeah, I mean, I don't mind answering it again. You know, we're seeing that there are gender differences, that there are differences in how women respond. So the case rate is pretty similar between men and women, but the mortality rate is much higher in men. And that might be linked to some kind of dose effect. So women have 2 X chromosomes. So generally one of those chromosomes is turned off and isn't making proteins. But for part of our immune system, it does. And so we kind of get double dose on some things. And that might actually be responsible for why women have better outcomes with this disease than men, just because we have more of the immune effectors that help defend us. As far as why people who are older, I'm trying to remember,
the question was people who are younger.
Yeah, younger people because younger people are exposed to a lot of things they've never seen before, and they're also developing their microbiome, so the organisms that naturally live on us, you don't want to get rid of them. You want us to create a good relationship with us and our healthy microbes. So they actually have a lot of immune modulators that suppress their immune systems to help them develop that healthy immunity. So there's some thought that those immune suppressors play a really important role in preventing that cytokine storm. And as you age, your body simply doesn't make enough of those anymore. So I believe that's certainly, and then of course, the younger you are, the less likely you are to have any of the comorbidities. You're not going to have been a smoker for 30 years. You might not have chronic pulmonary disease. You might not have heart disease. You might not be overweight. So there's other comorbidities that play a role. But certainly with the cytokine storm, it seems that the immune system of younger people is able to suppress a lot of the problems that we see.
And another one is, what are the status of, and in your opinion, what is the promise of monoclonal antibody drugs?
Yeah, monoclonals. Yes, so I'm going to try and talk a little bit about that. I'm going to talk about immunity, so I'll try and remember to pick up on that, and if I forget, remind me at the end. There was a really interesting paper that just came out that showed that there was really strong selection for just a couple of antibodies against the spike protein that provided protection. And so what you were seeing is that, so everybody, every human has our own particular arsenal of antibodies that we get from, you know, genetic reassortment of genes from our parents. And then they go under a second set of genetic reassortment that gives us our own unique antibodies. And then those antibodies are further sorted by response to the infection. So the likelihood of ending up with an identical antibody is essentially zero. And what we've been seeing is that people with COVID-19 have been developing exactly the same antibodies. So it could be that there is a monoclonal out there that would be really good at targeting it, if and that might be an approach as immunotherapy. Yeah.
Okay, last question, then we're going to go on. Can steroids be used to reduce the cytokine storm?
Mm-hmm. Yeah, that's one of the treatments, is to use steroids. Initially, when patients were coming in, the last thing you want to do with a viral infection is use steroids, because that suppresses the immune system, and you actually want the immune system to function to clear the virus. But now we know that's not the case. But there is a really specific timing that has to be used. If you start those steroids too early, you can actually increase the severity of disease. And if you start them too late, you've missed the boat. So yes, they work great, but you have to really know what you're doing in prescribing them to make sure you hit the target. That's a good question.
Okay, we're going to keep going now and let you do some more talking.
All right, so I want to talk about the vaccines that are in development, and I want to spend a little bit of time talking about immunity. So this is Omaha Beach from D-Day in World War II. So these are actually the Allied forces landing. You can see them getting their tanks and things like this. And so this is a metaphor I use for immunity when I'm teaching my students. So the idea is that, you know, we are France and our skin is the beach, and then we have an invading force that's trying to get inside and fight us. So in this case, we're actually the baddies. So we would be the Germans, and the allies would be the pathogenic organisms. So things like bacteria and fungus and viruses that are trying to get past our defenses and get inside. And the immune system is divided into two arms. One is called acquired, and the other is innate. And innate is going to be things that are basically structural. So like your skin is innate. So your skin is going to protect you no matter what. So you don't have to train your skin to recognize something foreign. It's just going to keep stuff out. The acid in our stomach has the same effect. And we also have cells that circulate. And if they see something foreign, they'll gobble it up. They'll recognize things on the surface of the cell as being foreign, and they'll run around and gobble it up. But they actually, that innate system, that's where some of the cytokine storm can happen, because you get a non-specific response. And the acquired is our trained immune system. So those would be, you know, your trained army, and those are our cells that make antibodies, and also the cells that kill virally infected cells. So I always like to put this picture up. This is the German defenses. So you can kind of imagine that this is our body's defense. So let's say you have a virus coming in here. You're breathing it in through your nose. You have all these nose hairs sticking up, and those nose hairs will filter out particles to try to stop the virus coming in. If the virus gets past those nose hairs, you have some thick mucus. I know no one listening here has ever picked their nose, but if you ever wonder why you have boogers, the boogers are there to basically, they're a sticky matrix. So if the virus goes flying past this sticky matrix, it gets stuck in the boogers, and then you can pick it out. If it gets past there, then it basically it will hit the barbed wire on the fence, and the barbed wire would be something like, you know, a cellular barrier or a skin barrier. I always like to think of the skin as the barbed wire. So here's the metaphor for our skin is the barbed wire. You can see that those might be German soldiers corralled on the far side of that. But anyway, you can see that it's keeping everybody on that side. And most of the times it works quite well. Skin keeps most things out. But if you get a scratch, what's going to happen is you might introduce, let's say this is a cat, you might be introducing viral particles or bacteria from the cat. And what's going to happen is you're going to get inflammation. So you can see there's a little bit of swelling there. The tissue's raised, it might get hot. And what that's doing is it's triggering an inflammatory response. And that inflammatory response sends out signals to the surrounding cells that something's cleared that barbed wire, something's got past that first line of defense, and we need to attack it and kill it. And because this is happening instantaneously, there's no time to train the immune system to defeat that. So you end up with people like this, right? So these are, you know, kind of locals who have guns. They don't really know what they're doing. They're going to shoot the guns off, and they're going to hope that they hit the right thing. Now, the trouble is these guys aren't trained very well, and they don't know when to stand down. And because of that, you can get an aberrant, you can get a misplaced immune response. So what they're doing is they're shooting their guns off all over the place. And by shooting their guns off all over the place, you're getting this dramatic inflammatory response, which is causing tremendous levels of damage. So they are basically the cytokine storm. So this is this non-trained portion of the immune system that are going around creating a lot of damage. So you don't really want that to be your primary mechanism of protection. So what happens is as soon as you get this invader, you start training your professional killers, right? So you hope that these guys show up. So you've got your trained army. They're really specific. They know what they're doing. They've got snipers. They only take out the right thing at the right time, and they can turn away and get rid of most of the pathogens in your body. And the reason that we are not sick all the time is because of these guys. They're trained and they have long memories. Some of the cells that are produced by the immune system that are trained can circulate in our body for 70 plus years, and they never forget. They always recognize a pathogen. If you're not too keen on this metaphor for war, I've got another picture of Chris Pratt there being the guardian of the galaxy, right? So whatever it is, you've got people that know what they're doing. So these are the trained cells. So these are the cells that we build up with vaccines. So we actually make vaccines specifically to train this set of cells to defend us. So this is this example of this. So this specific or acquired immunity, it's this trained army, and it's divided into two groups. One is going to be humoral, is what we call it. And that's what's going to be important in vaccine development. And that's where our antibodies are made, and they are made by B cells. The virus killing part of our immune system, these are the cytotoxic. Cyto means cell, and then toxic to kill. So these are cells that will actually go around, and if they see a cell that is infected with the virus, they will kill it. And you really want this to happen because in addition to making antibodies that neutralize the virus and stop it sticking, if these cells can recognize a cell as actually having the virus inside it, it can kill it before those virus particles are produced. So this is why it's really important to have vaccines, is because you can train these cells in advance to recognize invading pathogens and take them out. So we call it neutralizing immunity, in that you never know you've been sick. So, you know, if you get the flu vaccine every year, you've probably been exposed to the flu, but you never know it because these antibodies and cytotoxic T cells have protected you before you ever get sick. So it's a trained army, and it's trained in the thymus. So this is just a picture of the thymus. And the trick to our immunity is, how do you tell good from bad, right? So let's say I've got a virus particle out here, and I've got a cell here, all right? And this cell breaks up into little pieces that fly around the body that look maybe the same shape and size as a virus. And so how does our body know that this is good, all right, and this is bad? And we call this self. So anything that belongs to you is called self, and anything that's not you is called non-self. And if anyone ever tells you that they were abducted by aliens and that they were probed, they obviously weren't abducted by aliens, because aliens would want to know how our immune systems can differentiate self from non-self. So how can we see something as foreign? So if you think about it, if you've never been exposed to a novel virus before, so coronavirus is brand new, it's never been seen before, and yet you can develop immunity to it. So how does your body recognize something as foreign if it's never seen it? And what it does is it recognizes everything else as self. And what happens is your thymus actually presents every type of protein and tissue in your body. And your T cells come in through here. So these are T cells come in through your thymus and they come out the other side. And if they react with anything that belongs to you, they're killed. So they go in all nice and healthy, and anything that reacts with the human system is destroyed, and everything that comes out the other side will react with everything else. So you basically removed anything that can harm you, and it recognizes everything else is foreign. The only time this breaks down is when you see autoimmunity, and you get autoimmunity when you get cross-reactivity, because this system is not working very well. So that's how our bodies recognize something foreign, is they know what good things are, and if you recognize something good, you're killed, and everything else must be bad. So that's how we recognize non-self. And this is done by T cells, because those T cells mature in the thymus, and so that's why we call them T cells. So when you have an infecting virus, what happens is we have these cells, and they're called antigen presenting cells, or APCs. I like to think of them as part of the drain in a sink. So if you're, cleaning your dishes and you've got all those little bits of food particles that go down and they get trapped in the kind of the sieve at the bottom of the sink with the small holes, that's essentially what an antigen presenting cell is doing. It's basically constantly trying to see if there's any loose material out there. If there is loose material, it absorbs it and it presents it on the surface of the cell. All right, so imagine that's like the drain hole with the small holes in it. It's caught something in it and it presents it to the body. Now remember, T cells have been trained to only recognize things that are bad. So if a T cell recognizes this material as something that it's never seen before, it gets activated. So it's called a helper cell because it's activated. And it does two things. It turns on B cells, which make antibodies, and it turns on cytotoxic T cells, which kill infected cells. So this T helper cell has recognized something foreign, and it's nice and helpful. It turns on your antibody response, and it turns on your cytotoxic T cell response. It causes your B cell population to grow and make lots of copies of an antibody. Those antibodies will bind to neutralize the virus. It also makes these memory cells that allow your immune system to recognize that infection very, very quickly. So it can do it within hours of seeing it again. Cytotoxic T cells can actually recognize the virus particle on the surface of the cell. So our cells have the ability to break down pieces of the virus and present it on the surface, and that T cell can recognize that and kill that cell. So this is how our immune system works. I can teach you for 16 weeks on how exactly that works, but it is pretty fabulous. But anyway, if you want to make a vaccine, you need to make a vaccine that will activate both this part and ideally this part of the immune system, and it needs to activate them in ways that is beneficial to us. So there are some diseases that if you make too much antibody, they'll actually stick to the virus and pull the virus towards your cells, which for some viruses is not what you want. In others, the cytotoxic T cell might release the virus particle from the cell too early, and you might get more virus particles. So there's a real balance between getting this right, and that's why these vaccine studies take too long, because if they're done incorrectly, you can actually drive the immune system to make things worse. And obviously, if you've got something like a cytokine storm causing all kinds of problems, you're already kind of primed for potential problems. So that's why these vaccine studies take time, because you have to make sure that you're not making people sick, that you're not making the infection worse for them. Right now, I think there's over 100 vaccines in development. Some of them are whole virus vaccines, some are a viral vector. I'll talk about what that is. Some use a nucleic acid, so either a DNA or an RNA. Some are just proteins that are parts of the virus. And this other group here are using things like the BCG vaccine or polio vaccine to maybe stimulate the immune system to get a better response. A vast number of those are in North America, mostly in private industrial companies, some in China, Europe, and Australia. So the virus vaccines, these are whole viruses. There's two ways that they work. One is a weakened virus. So you've probably heard about this with polio, that you take a live polio vaccine. So when I was a kid and I had my vaccination, the doctor told me if I was good, I could have a sugar lump afterwards. So I was good and I got the sugar lump. And now as an adult, I know that that sugar lump was loaded with live polio vaccine. These are great vaccines because you don't need a lot of them, because they will get inside the body and they will make their own copies of themselves. They will spread around. You get a big amplification, you get a strong immune response, and you also shed the vaccine. So the reason the polio vaccine has been so effective is that if you're vaccinated against polio, you shed the vaccine and you can actually protect the people around you, and you basically are re-exposing people to the viral vector again. And you're also vaccinating people who maybe didn't get the vaccine. The other one is an inactivated virus. So this would be something more like the flu virus. So basically you grow a ton of the virus up and then you basically treat it with a chemical which kills it. So this works great because you get a really good immunity. The only trouble is you need so much of it because you need to get enough into the body to get the same kind of a reaction. So this vaccine here will self-amplify, make more copies of itself, and this one won't. What you get is what you have. So you might have to give multiple boosters of that. But they work great because they're taken up by the immune system. They're presented on the dendritic cells, so they're presented to the immune system and they stimulate a nice response. So these are great. There's not, I don't believe there's any, I guess there is one in, well, this one it's in New York, but they're in collaboration with India. A lot of these inactivated ones, I believe, are being produced in China right now. Okay, another one is a viral vector vaccine. So this is a completely different, well, it's a similar approach, maybe. So the idea is that you take a different virus, one that doesn't make us very sick, and then you put part of the coronavirus in it. So this, in this case, this is measles. So it's a measles, so it's highly infective. It spreads quite quickly, but it's inactivated. We've taken out a number of the genes which are responsible for making people sick, and that, you know, the encephalitis that you could get from it. Basically, those proteins are being removed. And what you've done instead is you've actually put in the spike protein of the coronavirus. So what happens, and this figure didn't catch it quite well, is that actually produces a lot of the spike protein on the surface of this vaccine. It gets in, it presents the spike protein. You develop antibodies against the carrier virus, but you also make them against the spike protein in the coronavirus. Another one is something that doesn't replicate. So again, this has an advantage because it makes more copies of itself. This one doesn't make more copies of itself, but adenoviruses are, if you've ever had kind of a sore throat, there's a good chance you've had an adenovirus. I think there's 80 circulating adenoviruses in the US. This one uses adeno5 as a good vector. And so what happens again is you put in the spike protein and it makes it on the surface, and then it goes through the process, your immune system recognizes it. Now, there's a big problem with these virus vaccines in that if you've already had an adeno5, so this is adenovirus 5, there's a good chance you've been exposed to it. Most people have seen it or been infected by it by the time that they're 5, like 20% of the population. So if you see this, your body is going to recognize it as foreign on all those ways that I've told you that it's trained to recognize it. And it actually starts to attack these viruses more aggressively than it does a spike protein. And there was a study in China where they used the adenovirus vector. So it was one of the very first virus vectors that was used. This is the same one that the folks in Oxford University are using in the UK, and it worked great as a vaccine unless you already had antibodies to adenovirus, and then you got much reduced immunity to the coronavirus. So it's probably going to be the first round of coronavirus vaccines because everybody can just kind of get loaded up with it, get a good response, but it's not going to be a long-term solution. But it'll be kind of like the polio virus, where we saw lots of iterations of the vaccine until we got to one that worked really well. Okay, so these nucleic acid vaccines are pretty interesting. The idea is that what you do is you shoot either DNA into the nucleus or RNA into the cytoplasm, and this gets translated from the nucleus, it gets translated into RNA and then into protein, and that protein is presented as foreign, or the RNA gets directly produced, translated into the viral protein that is presented as foreign, and you make an immune response. The advantage of these vaccines is that they're very quick to produce. You just basically have to synthesize a piece of DNA. It's really easy. It's something you can do. You know, we do it in my lab all the time. The trick is getting it inside the cell. So for these RNA vaccines, what you have to do is you have to coat it in kind of a soap bubble lipid membrane that will fuse with the membrane of our cells. So what you do is you basically take a hypodermic needle and you stick it into the muscle, and then a certain percentage of this material will be taken up by the muscle cells. The proteins will be expressed on the surface of the cell, and you get immunity. It's a bit trickier with DNA because it has to not only get through the membrane, but it has to get into the nucleus. And we do this all the time with bacteria by shocking them with like 2000 volts. And obviously that's hard to do. But apparently there is a nebulizer that you can buy, and you put it on someone's skin and it feels like a small electric pulse, and it will actually shoot it into the nucleus. Neither one of these methods has been used to produce a vaccine before, while the others have. But the nice advantage is that they're easy to scale up. To make enough doses of the RNA for everybody in the United States, I think they calculated they need about 240 kilograms. So that would be enough to vaccinate 400 million people. So figuring out how to do that's not going to be easy, but it's certainly an approach. And I think this is probably going to be second generation. I think this is going to be the one that long term we might need. And then the last one is something that's more common if you've ever had a vaccine against tetanus or whooping cough. It's a subunit vaccine. Basically, we just take a couple of the proteins from the virus itself and inject those into the body, and the system will take them up and present them, and you get an immune response. And this is kind of tried and true. The trick is getting enough expression of the proteins and purifying them in a way that will work. It also, you have to have a strong immune activator because the protein itself isn't going to make it happen. So we have to find something that accentuates the response. We call those adjuvants. And then a word of caution. So everybody's kind of like, well, let's get these vaccines out. Why aren't we doing this yet? So there's been some long-term consequences of the coronavirus infection, and this has been seen in young children. You may have seen this on the news. It's called Karasaki Syndrome. It's actually an autoimmune response where the body simply can't turn down production of antibodies, and you start to have antibodies that attack the vasculature. And so these kids are showing up with swollen toes and swollen fingers. Pretty high percentage, I think I wrote the number down here, something like where did I put it? 67% of children that present with this end up in the intensive care unit. And it's not just young kids. So Karasaki syndrome used to be kind of regular, what's the word I'm looking for, only found in children under the age of 5. So you'd really only ever see it here. But you can see we're seeing it in in young adults up to about 18 years. And it's only really seen about 5 to 6 weeks post exposure to COVID. And we're not sure what's causing it, but we do think it's a way that the immune system has responded to the virus. So there are some ways that poor presentation of the virus itself could cause problems in children. So, you know, the reason we go slowly with these vaccines, as I said, is to make sure that we don't make things worse. And the last thing we want to do is come up with a vaccine that works fantastic for anyone over the age of 20, but then anyone under the age of 20, it has a possibility of causing an autoimmune response. So I know people worry about vaccine safety, and you know, there's incredibly few cases. I think there's only 7 cases of vaccine litigation that have ever succeeded, and something like 400 million doses of vaccines are prescribed every year in the US, and that 7 cases goes back 50 years. So vaccines are one of the most safe drugs out there on the market. And the reason they're safe is because all these studies are done to make sure we're not going to make things worse. Okay, any questions, Debbie?
Yeah, we have just a couple. Let's see. It says, I'm asking about the promise of antibody drugs, not natural antibodies.
Yep, the monoclonals.
Yeah.
Yep. I think I answered that already.
Okay. What is the current best therapy if oxygen alone isn't sufficient? Anticoagulants?
Yeah, so they're doing a lot of anticoagulant drugs, I believe, and like big doses. Getting those blood clots down is the trick.
What
and the
would
immune
you,
response, turning down the immune response.
The last question is, what early version of a vaccine would you be comfortable with accepting yourself?
Oh, all of the ones that are out there. Yep, they all look good. I'm going to say checking to make sure that there's no secondary effect, but any of these that they're being produced, I'd be okay with. I mean, I don't know if I'd had an adenovirus, I might get sore throat if I get the vaccine, but it sure beats having coronavirus.
Okay, that's the last one for right now.
Okay, great. All right, so this is the last few slides. I know I'm dragging on again as usual, but in the last couple of weeks, some stuff's come up with bats, so I thought I'd address it here. So the big thing that people have seen in this report that came out from the Fish and Wildlife USGS, which is assessing the risks posed by SARS-CoV in and via North American bats, a decision framing and rapid risk assessment. So what was this document? So what it was an assessment of whether personal protective equipment would reduce the risks of human bat COVID infection if bats in North America could be infected with COVID-19. So it was a decision framing assessment. So it was basically, if it is shown that bats are susceptible, how are we going to handle that situation? So it was basically, should we make people wear PPE? What it was not was any indication of whether North American bats can get COVID-19. So I've had a couple of people tell me that a report came out that showed bats in North America can get COVID-19. They cannot. There is no, let me rephrase that. At this time, there is no data to suggest that North American bats are susceptible to COVID-19. There are ongoing animal trials at the National Health Wildlife Center. They have a colony of big brown bats, and they are in a P3 facility, so a high security, a biohazard security building where they are carrying out those tests right now. We do not have that data. That data is not being published. Right now, there is no information on whether North American bats are susceptible to SARS-CoV-2. This is some of the data that was published in that paper, and there was just a couple that I wanted to bring your attention to. So this is a assessment of the likelihood that if somebody is positive with SARS-CoV-2, how many bats of 1,000 bats encountered would they infect with SARS-CoV-2? So remember that, and this is a 10-minute exposure, right? So if they're exposed to a bat for 10 minutes, how many bats would they make sick? Now remember, COVID-19 is a human-to-human disease with a high R0 value, all right? So a high R0 of somewhere between 2 and 3. So it's highly infectious among humans. And we know that that susceptibility is 15% for 10 minutes of interaction. They managed to get values between 60 and 88%. So the likelihood of someone transmitting it to a bat as high as 88%. So this is a highly specific human-human disease with a high infectivity, and we're only saying 15% in humans, and somehow they predicted it was 88% in bats, even though there's no evidence at this point that North American bats can be infected. So that's the first kind of like, you know, red flag to me, like, I don't know what's going on. Now, this is an even bigger red flag to me. They've calculated, just kills me. They calculated the R0 value, so the infectivity for little brown bats, as somewhere between, I think this is like 0.2 and 6.5. So much, much more infection between bats than in a human disease. And one of the things I hope I've gotten across in the last 3 lectures is it's incredibly difficult to calculate R0. So how many people will get infected from a person? It's incredibly difficult to calculate R0. Even for coronavirus, where we have 7 million cases, we are still having trouble calculating the true R0 value. And without a single bat-to-bat infection or a single bat-to-bat transmission, they've actually calculated an R0, which is simply just not possible. So the way they did this was they actually put out a questionnaire, and they had 12 people respond to the questionnaire, and they were like, what do you think the likelihood of X is? And then they took all those numbers to come up with the value. And so it's an opinion of people. There's no indication of who these people were, what their background was. It said that they were bat biologists, virologists, and microbiologists, but we don't know anything about who those specific people were, that somehow they ended up with an R0 value, which you shouldn't even be asking that question. Anyway, so this is that. I was on the meeting where they actually announced these findings, and I was just pretty much in shock. So this is the question I asked. So alpha coronaviruses are very permissive viruses in North American bats. So most bat species in North America are infected with coronaviruses. They tend to be transient infections. So a bat will get infected. We don't know if it gets sick. It does make an immune response. It clears the virus. They're very permissive to alpha coronaviruses in North American bats, and alpha coronaviruses are much, much better at crossing species barriers and going through recombination events that allow species transmission than beta coronaviruses. So SARS is a beta coronavirus. The common cold, the one that we use a lot, is the 229E, is a common cold. So you've probably had that multiple times. It's an alpha coronavirus. So this is a circulating endemic alpha coronavirus in the US human population. And they did a study of something like 6,000 bats in North America, and they did not find a single incidence of a human circulating alpha coronavirus spillback event into North American bats. And 229E is an alpha coronavirus that's been in the human population for over 1,000 years. So it probably came to North America 500 years ago, and there's no evidence of it circulating in bats. So let's say humans in North America have been interacting with bats for 300 years. There's not been a spillback event for a known alpha coronavirus into a North American bat with a circulating permissive virus in 300 years. But they think that there's an 88% chance that could be for this SARS. So anyway, this is just more evidence that, and I did ask about this, and their answer was we only looked at beta coronaviruses, but then they don't have any data on beta coronaviruses. And then this is just a study that came out. This is some molecular phylogenetics. So I don't expect people to know much about this unless you've got a background in evolutionary biology. But basically, somebody compared all of the known SARS coronaviruses, including the bat SARS-like viruses, the closest relatives to the current outbreak. And it looks like there's been two evolutionary events. One that allowed the virus to jump out of the bat population, and then a second event, which was a gain of function, which allowed this spike protein to gain an additional cleavage site. So there's a lot of evolutionary evidence that there's been a recombination event, so a genetic shuffling that happened somewhere before 1992 that allowed the virus to escape the bat population in China, and then another genetic reassortment that occurred somewhere around 2009 that allowed this virus to spread from human to human. So this may have been, this period here may have been when we were looking at an intermediate host in China. And then this is some more data. There have been SARS-like beta coronaviruses identified in Chinese bats. They've only been found in two genera of bats in China. These are horseshoe bats. There are over 100 species of bats in China that swarm together and commingle, that have very large ranges. This is the range of those horseshoe bats. This is where SARS coronavirus has been identified. So it hasn't spread elsewhere. It hasn't spread outside the habitat of this particular genus, and it's not spread out of this genus into other species, even though there are other species sharing the same bat habitat. So these SARS-like coronaviruses appear to be restricted to these genera. So it's a very, very narrow host range. And somehow they're jumping out of here, maybe into animal populations. There is a lot of farming, a lot of livestock, these wild animal breeding areas for markets, and they are near and around caves. So there's lots of opportunities for these viruses to jump into an intermediate host. Now, that's not to say there couldn't be a human host, and these recombination events did happen in a human host, but the easiest explanation is that there was an intermediate host. And then finally, somebody brought this paper to my attention. This is a paper that showed infection of bat cells with the SARS-CoV-2. So these are called organoids. So basically, it's a culture of individual cells, and it's very hard to get them to grow. So what you do is you kind of grow them as a blob. And then what they've done is they've exposed this blob of bat cells. These are gastrointestinal cells. So these are gut content cells from the horseshoe bat, so the same bat that the SARS-like virus is seen in. They've taken this blob of cells and they've infected it with the SARS coronavirus, and they've painted that SARS coronavirus green in this panel. And you can see that some of the cells, just a couple, have become infected with the coronavirus. Now, what they've done is they've actually poured the virus over the surface of these cells. And remember, this is a horseshoe bat, so this is an ancestral cell line for the virus. And they've shown that there is infection, but it's 1,000-fold less than if you did the same experiment using human cells. They also stained to see if the ACE2 receptor is present on the surface of these cells, and they did find an ACE2-like receptor, but it's only 80% identical to the human one. So it's possible that if you have a high enough amount of the virus present, it will get taken up, but there's no indication of whether this was a specific receptor interaction, or it's basically that there's fusion of the virus with the cell surface that's being taken up. So this is in a culture. It's not an animal, It's not exposing an animal in one cage and showing that you have virus particles in the air that are infecting the other animals. It's also intestinal cells. There's no indication that if a human was coughing, that virus could end up in the intestine through that route. And then the last one is permissive hosts. There has been a lot of work on animal species that are permissive to infection and animal-to-animal infection. So cats have been permissive to infection, only under very specific conditions, but it can happen. And it's been shown that that can happen for an aerosol, so basically through the air. Ferrets, it's the same thing, is that they are susceptible to infection and they can actually spread the infection between populations of ferrets. And hamsters, so it's a pretty good animal model. Hamsters will actually get infected and produce an aerosol that can infect other hamsters. Other animals that have been challenged and actually do develop the virus are dogs, pigs, chickens, and ducks, but they haven't been able to spread it to other animals. So basically a single animal, when exposed to a high dose of the virus by pouring it up its nose, will actually make copies of the virus, but it doesn't produce a permissive infection. So that means it doesn't make enough virus or release it in a way that can infect. And you'll notice that right now there are no bats on that list. All right, so I'm going to finish up there. This is my last slide. I didn't mean to have a rant. I decided to change it up a little bit from the last slide I had up. This is a new book by Paul Rabidan. He's the guy who did the molecular evolutionary history of the beta coronaviruses. He's a really interesting guy. He was writing so much about coronaviruses, he decided to just put it all together in a book. And so that's his book that's available on Amazon. And he's a great guy. I recommend it. And then the other is that conspiracy theories on Facebook have kind of died off a little bit because there seems to be a lot more that people are focused on. And I just wanted to, this was a cartoon, I can't remember where I saw it, where, you know, we're starting with the data, we synthesize the data into information, we take that, we try to build knowledge and then insight, and that leads to wisdom. And that's how science is, and that's why science takes too long. And then this is the conspiracy theory, where you basically find a bunch of random facts and string them together, and all of a sudden Bill Gates has created a virus to inject us with microchips as opposed to just using phones. Anyway, that's it. I'm going to finish there.
Okay. Well, let's get to the questions. All right. Oh, one person said the same thing I thought is, you are not dragging on. We are all spellbound.
Oh, cool.
So, well, seriously, you've got a great attention statistics here. It says a couple of doctors in Italy have said that it seems the virus has become less virulent and less deadly. Is it possible for this to actually happen?
Yeah, it's how viruses evolve. They always evolve with selection. So they want to transmit more, and they don't want to harm the host. The more you harm the host, the less likely you are to be transmitted. So they always tend to mutate towards being less virulent. Now, I think I've heard about that study, and it was anecdotal, and they didn't have any actual data and metrics. They had basically seen some patients, and their cohort that they were seeing didn't seem to be as sick, but there was actually no data to support that. So it's based on evolutionary biology. It should happen that way, but that right now that's anecdotal evidence. But yeah, it hopefully it'll go that way.
Mm-hmm. Okay, let's see. Now the questions are coming in so fast. Mm-hmm. Are self-replicating adenoviral viral vector or RNA vaccines more ideal for first-line vaccines for materials manufacturing reasons?
Yeah, that's an interesting one. The adenovirus is easy because that's actually a vector that's been used for doing gene therapy. So there's precedent of using that in humans, and there's lots of different ways of getting that in. And it's basically a plug and play. I mean, it's the kind of molecular tool that you'll have on your shelf, and you can just basically pull down and add all the constructs together, stick it in a cell, and it will make your virus. So that's pretty easy. I mean, that's a pretty obvious first approach. The only issue with that one is that for long-term immunity, if you've been exposed to adenovirus and you've got adenovirus antibodies, you might not make as strong a response. And certainly the strength of the immune response seems to be critical. The people that aren't clearing the virus don't seem to be making a strong immune response. So I think that there's going to be so many different kinds, and everybody's going to be like, I thought it was this one, I thought it was that one. Think about the polio vaccine. It took us 50 years to find the perfect one. I mean, we're going at breakneck speed, and I imagine there will be some iterations. The first ones will protect us. They might not give us, strong lifelong, but they'll certainly help save our lives. And then as things develop, we might get more refined and better forms of the vaccine. I think the messenger RNA one's good. Theoretically, the only limitation with that one is how you make enough messenger RNA to be able to vaccinate all those people.
Okay.
It's a good question.
Yes, it is. What is the chance that cats, ferrets, or similar species are an intermediate host?
Yeah, it would be nice to know that. I think that there seems to be some commonality there. I'm not a mammologist, so I'm not very good at this. I kind of had my money on cats, actually, because of the markets. When you go to these markets, there's tons of cats and cat-like things there. So I imagine they might be clues. There's certainly a lot. There was one study of a fruit bat in African fruit bat that was done, but they injected a ton of the virus up its nose, and they didn't control for contamination. So we don't know, but yeah, there's actually, I was going to put it into the talk, but I didn't in the end because I wondered if it would be being tried to be too controversial. But there's actually some studies that think that the reason we think most of these viruses come out of bats is because bats are oversampled. It's really easy to catch bats in the habitat because you just, put a mist net up over cave entrances and you can just grab these things out of the air. And so bats have been highly oversampled compared to other animals. So there's some people that have argued that, blaming bats all the time is more about oversampling than anything else, because civets have a coronavirus, SARS-like coronavirus. The pangolins have a SARS-like coronavirus. Certainly it's ancestral in bats, but that doesn't mean there aren't other coronaviruses that maybe have been near misses and we should be concerned about. But I certainly think it's unfortunate that the National Park Service is requiring PPE and N95 masks for anyone around bats, but you don't have to do that if you're around ferrets, and we know ferrets are permissive. So it's a bit kind of like, and then hear people telling me like, oh, there's a paper came out, it's like really infectious in bats. It's like, oh my God.
Okay, well, the next one is has to do with minks. It says, what about the minks in the Netherlands that have been infected, that have infected humans?
Yeah, I heard something about that. I've never seen it. I haven't seen a published paper. So that's why I limited it to just that list where I was able to actually see. And it could be that's just a paper I've missed. There's been a lot rolling around. I do vaguely remember hearing something about that. But I can't remember if there was some suspicion about whether the humans had really caught it that way, or maybe there was an infection that hadn't been, like a exposure that hadn't been recorded. So I honestly can't. I'm just guessing at this point. I do vaguely remember hearing about that, but I haven't seen anything published.
Okay. Another one is, what is the probable time frame of elevated temperature and also asymptomatic spreaders, and why are we bothering to even screen for temperature?
So temperature is one of the things that everybody gets. I think it's over 90% of patients get a temperature. It could be a mild temperature, so mild that you don't even notice you've got it. But if you have a thermometer, you can spot it. So one of the things that I've been recommending at my university is that we have temperature stations at every entrance. And you basically, if someone shows up with a slightly elevated temperature, you just ask them to come back tomorrow. And if it's high again, then they need to go see a doctor. So the signs can start as early as 1 to 5 days. Most suggestions are that so this is a really infectious virus. So from it infecting a cell to making 10 to 40,000 copies of itself and then leaving the cell is 4 hours. So if you get one virus in you and then it replicates, that's tens of thousands of your cells that can be infected. And then they in turn produce enough virus to infect tens of thousands of other cells. So it can be as short as a day. So there is data to suggest that it can be as short as a day to go from being infected to showing signs and symptoms and shedding the virus. The average is, I think, 2 to 4 days, which is really, really fast for a virus. But this virus has to do that because our immune system, those antibodies and those T cells, they're ready to go to 4 to 5 days. So this virus has to get in and it has to do its thing and it has to start moving, because as soon as that trained immune response kicks in, then the virus is going to start getting wiped out.
So how does a virus know to do these things?
It's not sentient, it's evolution.
Okay, when they cut genes from a virus to weaken it, do we know which genes to cut, ones that make it deadly, or is that one thing that contributes to what takes so long to formulate a vaccine that we need to learn what genes to cut?
That's a good question. So we know an awful lot about adenovirus and measles. We've been studying them for years. The adenovirus vector has been, as I said, being used a lot in biology to deliver gene therapy, and certainly measles. So the way that we do this is actually you can set up these cultures of cells, and what you can do is you can actually go in and knock a gene out and look at how the virus functions. So it's actually, viruses don't have a lot of these genes. I think, I'm trying to remember, coronavirus has got like 23 or something. It's not many, and it's a fairly big virus. HIV's only got 9. And so what you can do is you can actually tinker with them and see what the effect is. And you can look at how they make copies of themselves, they bind to cells, they release from cells, and then you can take those into animals and you can actually, see how the immune response in the animal works. So a lot of this groundwork has been done. Certainly a lot has been done for the adenoviruses. So it's really easy to know what those changes are and to do them. The trick is you have to basically build your DNA blocks, then you've got to get that into the cell, then you've got the cell to make virus, then you've got to purify the virus particles, then you've got to assay them, so you've got to see if they're functional, then you've got to make sure you've got exactly what you're looking for, and then you move into the animal models, and out of the animal models, then you can start moving into humans. So that's what slows it down. The actual building part, I mean, if you're in a good lab that knows what they're doing, that does this on a regular basis, they can probably build that virus in 2 weeks. It's all the other things that take a long time to do.
All right, let's see. That's it. All right. Any other questions, please speak now. If you think of questions later, go ahead and email us, and I forward them. You can email Hazel, or you can email me. I forward them all to Hazel. But if there are no more questions, because you've been answering quite a few, we're going to go ahead and close that for the night. And Hazel, you're amazing for keep coming on and giving us all this great information. We are very grateful.
Things on Facebook and getting frustrated, and then seeing people publish things that say that bats are 88% likely to get the virus from a human. So, yeah, I just get motivated.
Want to knock some sense into them, right?
Yeah, it's not my place.
All right, everybody. Oh, it says, thank you very much. Well done. Stay angry about dumb people. I think I sent you this one of an elderly gentleman quite old. And one man said, "How did you get to be so old?" And the older man said, "Because I never argue with stupid people." And the other guy goes, "I don't think that's right." And the elderly gentleman said, "You're right."
Sounds good.
So
I should
arguing with stupid people just doesn't get you anywhere, because you can't educate them if they don't want to be educated.
yeah.
All right. Everybody, thank you. Hopefully, sometime soon, we'll be able to go caving. We're just hoping that things get calmed down enough that we can actually get back underground. So anyway, everybody, thank you for joining us. We want to thank all of those that are first responders, medical professionals, researchers, anybody that is working on this this incredibly difficult situation. We want to thank you for all your dedication and your sacrifice. And we will, this will all be recorded, start on the NSS website as usual at caves.org. You can download, you can watch it online. Please share this with whomever you'd like. This is great educational information. It will also be posted on the NSS YouTube site, so you can send people a link to that also. And Hazel, again, thank you very much for your time and effort.
You're welcome, Debbie.
And for Derek and his helping us edit all of these things and getting everything in working order. All right, everybody, take care and cave safely. We will talk to you sometime in the future. Have a good night.